Now, each one of is to endow each one of these arrows with molecular and quantitative information that allows us to reach a predictive understanding of cellular decision making in development. Okay, namely, could I predict a developmental phenotype from knowledge of this network topology? So to be more specific, we don't try to, in the beginning, to do all the arrows at once, but we focus on individual arrows or small groups of arrows, and we ask, can I predict how the arrangement of binding sites on the DNA dictates the so-called input-output function, the functional relation between the output level of gene expression and the input transcription factor concentration. Okay, can I do this quantitatively in terms of quantitative phenotypes? And can I do this so well that I can predict how this shape will change as I tune the number, placement, and affinity of transcription factor binding sites on the DNA? So how do we pull this off? Well, what we try to do is to engage in the same dialogue between theory and experiment that is the hallmark of the physics approach. Okay, and by that I mean that we grab these biological qualitative models of biological function that you find often in textbooks, in biology textbooks, and we turn them into precise mathematical statements that predict, for example, the input-output function. And then we go into the lab and we measure input, output in order to test the theoretical prediction. And the idea is that this is a dialogue, this is a cycle. Theory is there to make experimentally testable predictions. Experiments are explicitly designed to test those predictions and to inform the next round of experimentation, of theoretical modeling. And what I'm going to try to show you today is that how this dialogue is not just interesting as a means to reach predictive understanding, but also can be used as a microscope to glean into molecular mechanism, to see things that you couldn't even see with the fanciest super resolution microscopes. Okay. All right. So how do you do this? Well, so just to give you a little bit of a sense of how I got to this place, during my PhD, I tried to ask these sort of questions, but in the context of bacteria, in the context of E. coli. For example, we developed models that predicted the level of gene expression as a function of the number of repressor molecules and as a function of the number, placement, and affinity of those repressor binding sites. And then we developed technology. We went into the lab in order to test those predictions. And to a large extent, what we showed is that we can predict these input-output functions with only a few or no free parameters. Okay. So then for my postdoc, I decided, let's try to see if we can pull this off in the context of a multicellular organism. And I decided to work on the fruit fly because it has like a fantastically well-mapped gene regulatory network. You can you know a lot about the molecular players, so you can connect molecular players to the body plan. And so what I focused on, and I'll show you some of this today, is on developing technologies to be able to engage in this theory experiment dialogue. And today, now in my lab, we work mostly on the fruit fly, and that's going to be the biggest, the main topic of today. But we've also been expanding, trying to reach a predictive understanding, not just of fruit fly development, but also of different molecular processes in the context of zebrafish, Arabidopsis, C. elegans, and stem cells. Okay. But let me be more specific. What do I mean about being able to reach a predictive understanding of multicellular organisms or of their development or enabling this physical biology approach in the context of animals and plants? So there's a few challenges to realizing this. And one of them is that you need technology to measure the very same input-output functions that we're trying to predict. Okay. Second, you also, I told you about grabbing the qualitative models, these cartoon models, and turning them into math. But sometimes we don't even know the cartoon model. For example, you can say we know things about when repressors bind or activators bind and unbind from the DNA, but we still know surprisingly little about what they actually do once they're bound to the DNA, how they actually work at the molecular level. And you need that in order to write theoretical models. And finally, we need to figure out how to derive and test theoretical models of gene regulation. So these three challenges represent the three main research threads in my lab over the last 8 years. Okay, so we've had to develop technologies to light up the processes of the central dogma to visualize regulatory control in single cells of multicellular organisms, something you couldn't do before, and without which we couldn't really measure these input-output functions. We also used physics as a microscope for in vivo biochemistry to uncover the molecular mechanism of a transcriptional control so that we can write theoretical models. And finally, we put this all together in the service of realizing physical biology of developing embryos. And we've had great fun thinking about along all of these lines throughout the last few years. And today, what I'm going to do is tell you about a story that will give you a glimpse of how each one of these works in the service of realizing physical biology of developing embryos. Specifically, what I want to do today is to figure out what is it that I need to know in order to be able to predict this input-output function. So the input concentration of transcription factor together with the arrangement of binding sites on the DNA dictates the output level of gene expression. And already you see that maybe this cartoon is a little bit too simplistic. Like instead of looking at the concentration of the transcription factor throughout the embryo, maybe what I need to do is to know what are the dynamics of this transcription factor in the nucleus. How do they bind and unbind to the DNA? And how does that binding inform the transcriptional dynamics of individual genes? And finally, how do all those cells transcribe in a coordinated fashion in order to give you an output pattern of gene expression? So today, what I'm going to show you is how we use the theory-experiment dialogue in order to shed light on each one of these steps between input and output in development. Okay, and the story I'm going to talk to you about today was mainly spearheaded by Megan Turner and Nick Lammers in my lab with the help of Augusto Berrocal, Emma Liu, and Simon Alamos. And it's also the result of a great collaboration with labs of Chris Wiggins at Columbia, and the labs of my colleagues Mike Eisen and Xavier Darzac at Berkeley, and a bunch of really great collaborators like Baki Galstian, Mike Stadler, Jenna Haynes, Thomas Graham, and Mustafa Mir. So, and the talk is going to be divided in 4 very short parts. The first thing I'm going to do is introduce you to the technologies we have to develop in order to engage in the theory experiment dialogue. Those technologies are going to allow us to look at transcriptional dynamics in single cells. And what we're going to see is that transcriptional dynamics doesn't proceed continuously, but it's characterized by these bursts. And we're going to find that transcription factors can control the bursting dynamics in order to modulate levels of gene expression. We're then going to ask, how do transcription factors implement this function at the molecular level? And so we're going to start going through multiple cycles of theory and experiment trying to answer that question. And every time we go through one of these cycles, what we're going to find is that we're going to break the model. And ultimately, we're going to converge on a discovery, which is that transcription factors are not uniformly distributed throughout the nucleus, that they can be present in clusters. And when you acknowledge that these clusters exist, you can come up with theoretical models that can recapitulate the bursting phenomenology, at least to some degree. Okay, so let me get started. Before that, are there any questions? This was just no. Okay, all right. So then I'll just get started. I'm sure there'll be more questions in the next few parts. Okay, so let me just get started. And the main player we're going to be thinking about today is probably the most understood pattern of gene expression in development, which is the striped 2 of one of the 7 striped genes called even skipped. Okay, so even skipped, you see it here with measurements of in situ. This is accumulation of RNA. We know that even skipped is created by 2 activators and 2 repressors. And initially, only the activators are present along the embryo, and which generates a broad domain of activation of even skip. But then the repressors come from each side, and they restrict the domain of activation to this little stripe that you see here. That is about 6% of the embryo length. And we don't only know the identities of the inputs of the activators and repressors, we only know the arrangement of their binding sites. So once again, it's a pretty cool state of the art to get started with. And yet, everything we know about the most understood pattern of gene expression in development comes from data like this, where basically what you're measuring is the accumulation of RNA in the cytoplasm, okay? Which means that this data is completely silent on what are individual cells doing in order to generate this pattern. By that I mean, you could have cells in the middle of the stripe expressed at a higher level than cells on the sides of the stripe. You could also have just more cells in the middle of the stripe expressed at a higher level than cells on the sides of the stripe. Or you could have any combination thereof, right? So if you're going to get started in trying to write governing equations, the first thing to figure out about transcriptional control is how this control is realized at the single cell level, okay? And to do that, basically what I'm telling you is that we need to measure these input-output functions at the single cell level in real time, okay? But there was a problem when we started thinking about this. And that problem is that, typically imagine that you want to look at an activator concentration. Typically, you would fuse it to GFP. But the dirty little secret with GFP and fluorescent proteins in general is that the moment it gets translated, it's dark. And it takes some time for it to mature. It takes on the order of 40 minutes for it to mature in worms and fish and frogs and flies. Okay? This wouldn't be a big deal if it wasn't for the fact that the transcription factors, the activators and repressors we want to look at, only live for 10 minutes. So by the time the fluorescence kicks in, the action is over. Okay? What this constrained the field in general is to looking at pictures of dead embryos. What I mean by that is that people would grab embryos that were laid at different time points in development. You don't know exactly what time point. Those embryos would be stained against some protein, some RNA, and then you would make a fake movie where you're morphing through images of dead embryos. So it's an amazing, amazing technology, and people got a lot of mileage out of it. But clearly, if you want to measure transcriptional dynamics in an individual cell in real time, this is not good enough. Okay. So over the last few years, we've developed technologies to measure the input, to measure the output in real time in single cells of living developing animals. And what I want to show you in a little bit of detail is the technology that we developed in order to measure the output transcriptional dynamics in single cells. And this is the so-called MS2 system that was originally invented by Rob Singer to look at transcriptional dynamics in single cells, and that we had to re-engineer in order to make it work in animals. And this was work that I did when I was a postdoc in Thomas Greger's lab, and similar work was also done in Natalie Dostatny's lab at the same time. And here's the idea. You're going to grab a sequence that you're going to put in the gene that you care about, such that when that sequence gets transcribed, it forms a loop. That loop is recognized by a binding protein fused to GFP, such that every nucleus in your embryo will have a little spot of fluorescence that tells you how many polymerase molecules are actively transcribing the gene. Now, note that the GFP was produced way in advance, so it's mature. What you're looking at is a localization of GFP, and that's much faster than the maturation process. Okay? So this is what it looks like when you put it all together. In red, you see DNA, and in green, you see the RNA signal. So you see how first we have this word activation domain that I told you about, and then as development progresses, the green dots are going to be restricted more and more into the little stripe that I told you. Okay? So this is the first time anybody could see transcriptional dynamics in single cells of a multicellular organism. Not only that, one of the cool aspects of this is that we're looking at an in vivo single molecule experiment, because each one of these dots reports on the transcriptional decisions made by an individual DNA molecule. Okay? So I just showed you how we can measure the output. Now we're zooming into that stripe, and you see how it becomes narrow. So that's the output. We've also developed technologies to measure the input. Like here you can see one of the repressors that comes from the side and restricts the fluorescence, restricts the transcriptional activity of the stripe. And we can do this in the same embryo in real time. So with these technologies, we can measure input and output, or input-output functions in single cells in real time in living multicellular organisms. Okay? And these technologies have helped enable quantitative developmental biology writ large, and with many different labs using them now to answer their own questions in the context of fruit fly development. And also we've been expanding them to, by sharing the technologies, and also in the context of collaborations, to plants, to fish, to worms, and to stem cells. And as I said, many of them in the context of collaborations. And the reason I'm mentioning this is not to boast or anything like that. It's because I just want to get the message across that we're committed to creating and nurturing a vibrant community of quantitative developmental biologists. So we share reagents and code before it's published. We regularly host people in the lab, the visitors that want to learn how to implement the technologies, how to do the data analysis, how to do the microscopy. So if anybody's interested in these sorts of technologies, feel free to shoot me an email after the talk, and I'll be very happy to talk to you. Okay. All right. So I just showed you how now we have technologies to answer for the first time what are individual cells doing over space and time in order to create this pattern, this macroscopic pattern of gene expression. Okay? And what I want to do next is to show you how, when we use these technologies to zoom into the transcriptional dynamics of individual cells, we're going to find that these individual cells are transcribing in a bursty fashion. And the transcription factors seem to modulate transcriptional dynamics by controlling the parameters of bursting. But before I proceed, are there any questions about what I just showed you? Just unmute yourself and ask, or something like that. No? All right. Okay. Well, I'll just keep going then. Okay. So what do? We zoom into one of these little dots, and we measure the number of RNA polymerase molecules as a function of time. And what we find is that the promoter is not transcribing all the time. The gene is not transcribing all the time. It's not on all the time. Instead, it looks like transcription is characterized by these episodic bursts of gene expression. Okay? And you can think about this transcriptional bursting in many different ways. One of them is like, one thing you could ask is, well, clearly this sort of on and off has to be more noisy. So how does development buffer the noisiness coming from these transcriptional bursts? And that's a great question. But what I want to focus on today is a different question, which is, can we use bursting as a window into the molecular mechanisms of transcriptional control? Okay. And let me just say that bursting is not something that is isolated to flies or to a particular gene. It seems to be the rule rather than the exception. We keep finding it all over the place in animals, but also people have seen them in bacteria, in yeast, in cells in culture. Okay, and the simplest model that one can use to project this bursting phenomenology is a so-called two-state model, where you posit that the promoter is going to be transitioning between being in an on and an off state in a stochastic fashion, and only when in the on state it will transcribe. So in this context, you could, if you look at the RNA polymerase loading rate as a function of time, you could define a burst frequency, a burst length, or a burst duration, and a burst amplitude. But now that we know that the gene responsible for making the stripe is bursty, the question that we wanted to ask of how individual cells make the stripe becomes a little more nuanced. What do I mean by that? This is measuring the accumulation of RNA or the mean RNA production. Okay, and if you acknowledge that there's bursting, this mean RNA, mean rate of transcription is given by this equation here, where you have a term that has to do with the fraction of time that the promoter spends in the on state, and a term that tells you what is the transcription when you're in the on state. Okay, and in the context of this model, then you can make a stripe by increasing burst frequency in the middle of the stripe, by increasing burst duration or decreasing K off in the middle of the stripe, by increasing the burst amplitude in the middle of the stripe, or any combination thereof. Like, could be some combination of all these three strategies. But the point is that if you want to answer the question of how the individual cells regulate their transcriptional dynamics in order to make a stripe, you need to be able to measure burst frequency, duration, and amplitude as you move along the embryo. And this would be trivial if you could measure the rate of polymerase loading as a function of time. You would just see it coming on and off. You would measure K on, K off, and R, the three parameters of the two-state model, and you would be done. The problem is that our technology doesn't measure the promoter state directly. The promoter state is hidden from our view. What we measure is the number of polymerase molecules on the DNA that are actively transcribing. And these two quantities are not the same, but they're related. And you need to do an inference in order to relate the two. Now, this is easier said than done. For years, we were sitting on terabytes and terabytes of data that we didn't know how to analyze. And only when we started thinking with Chris Wiggins about this problem, when we started repurposing so-called hidden Markov models, did we figure out how to do this. Okay, so I'm not going to go through the math right now. We can talk about it later. But let me show you what this can do. Okay, so here on the top, I'm showing you the number of polymerase molecules transcribing the gene as a function of time for an individual cell. And sure enough, you can tell me that this is bursting, but you would have a hard time telling me when a burst begins and when a burst ends. And now we can run our inference to infer the most likely state of the promoter at each time point. Was it on? Is it off? Okay, we can do this for every nucleus throughout the embryo. So here I'm color-coding the nuclei, depending on whether the inference determined that the promoter was on or off at each time point in development. And what I wanted you to notice is that even in the middle of the stripe, where you have a lot of fluorescence, a lot of transcription, the promoters are still turning on and off. There is no such thing as being on all the time. So what we can do now is move along the embryo and ask at each position, what is your burst frequency, your burst amplitude, your burst duration? Okay. So why did I want to ask this question? Well, remember, because we're trying to figure out which out of the 4 transcription factors that dictate the expression of this stripe, are they controlling, which parameters are they controlling, K on, K off, or R? So we move along the embryo and we measure each one of these bursting parameters. Here, I'm showing you the rate of polymerase loading, the burst amplitude, as a function of the position along the embryo. And what you see is that it doesn't change much, even though there's a stripe here in the middle. If you look at burst duration, there's also not much of a change as you move along the embryo. But so this tells you that neither of the 4 transcription factors talks to the K off, talks to R. What we find is that the burst frequency is regulated. It's modulated in a stripe-like fashion, just like the ultimate stripe that you get. But this is interesting because there's 4 transcription factors, and they're not related necessarily, and they all seem to be acting through the regulation of burst frequency. And this seemed to be consistent also with what people had measured in single cells. So for a while, I thought, okay, there's some unity of mechanisms here because transcription factors are constrained to work through just one biochemical pathway. But then we started looking at more genes, and we found examples of sets of genes where, for example, frequency and amplitude is being regulated. Other examples where duration and amplitude is being regulated. Other people have also found examples of frequency and duration regulation. So while there seems to be unity, there also seems to be a little bit of a zoo of regulatory strategies out there. And in general, what I want to say is that we have become pretty good at determining which bursting parameters are under regulatory control. What we still have no idea is how this control is realized at the molecular level. Honestly, we don't even know what a burst is, let alone how it's being regulated. Okay? And clearly, in order to answer those sorts of questions, you're going to have to go beyond just measuring the output and know something about what the transcription factors are doing when they bind to the DNA. So just to be specific, imagine that maybe a transcription factor needs to be there for the whole duration of the burst, or maybe it just needs to be there at the beginning of the burst to prime the burst. Okay, so these are just two not mutually exclusive and not exhaustive scenarios, but I just want to just to give you a sense of like how you actually need to measure both input and output in this case. Okay, so I just showed you how when we zoom into individual cells, those cells seem to be transcribing in just discrete bursts of expression. And also I showed you that transcription factors seem to regulate the transcriptional dynamics by modulating some set of parameters, where it's frequency, duration, and amplitude. Okay, any questions so far?
So we have a couple of questions in the chat.
Right.
Claudio Franco asks, do you know how many mRNA molecules are transcribed in each GFP dot?
Yes, yes. In fact, if you look at the,
I
so
guess
we
it was
can
on your slide.
do all sorts of different calibrations, but you can see here we actually know the number of polymerase molecules that are being transcribed. And what I can tell you is that if you have on the order of 60 polymerases being transcribing, that means that the polymerases are one after the other. There is no room in between polymerases, which is basically the speed limit of transcription.
And the second question from Ulrich Kaiser is mRNA expression a quantitative indicator for protein expression in this system?
As far as we can tell, it is. As far as we can tell, it is. Though there is room, and people think there is room, and people think they have measured this to regulate the translation rate in space and time. So yeah, in principle, we believe that this is a faithful reporter of the rate of initiation, but then there's a question of can you use that to predict the protein? And this is something we're very interested in and we're working on. People have found evidence that some of the translation rate is regulated in space and time. So the answer is not going to be always that it's a linear function, one of the other. Okay.
Okay, great. Those are all.
All right. Well, keep them coming. If anybody wants to ask anything, also feel free to unmute yourself. Okay. All right. So what I want to do now is put on my theorist hat and try to ask, how can you come up with, what are the sort of molecular mechanisms that one could come up that would explain the bursts and their regulation? Okay. And the simplest model one can think of, and this is a model that seems to work in yeast, according to Dan Larson and Tineke Lancer and a few others. Simplest model you can think of is one where you have a binding site for your activator, and the moment the binding site is occupied, now you're bursting. Okay. In this simple model, what you would predict is that the burst frequency is given by the activator concentration, because burst frequency is the same thing as the activator binding rate, right? And activator binding rate increases with the amount of protein. There is also another prediction this model makes, that burst duration remains constant, because burst duration would only be dictated by the residence time of the activator on the DNA, and that should be independent of the activator concentration. Okay, so but what this kind of tells you is that if you want to actually test these models, you're going to have to be able to compare burst dynamics and binding dynamics. Okay, and so far everything I've shown you has been done with laser scanning confocal microscopy, which is amazing, but does not have enough signal to noise to look at individual activator molecules binding and unbinding. Okay, but fortunately, my colleague Eric Betzig had just invented this so-called lattice light sheet microscope that allows you to shine light on using a very thin sheet of light throughout the embryo, dramatically increasing the signal to noise. And then Xavier Dalsac, another Berkeley colleague, built one of those in the lab, and we started putting embryos underneath, under the microscope. And so let me show you a little bit what the data we're gonna stare at looks like. Okay, so I'm labeling one of the activators with GFP. Okay, and I'm going to look at individual molecules move around. Some of them are going to move a lot. Some of them are not going to move that much. Okay, we're going to say that the ones that move a lot are going to be not bound. The ones that are bound are going to be the ones that don't move that much. And obviously, we don't have infinite temporal resolution. We have some exposure time that kind of, and what it means is that the unbound guys are gonna, their fluorescence is gonna be very diffuse, and the bound guys are, their fluorescence is gonna be very concentrated. Okay, so I can look at bright dots and learn something about who's bound. Okay, so this is what the data looks like. This is again on the lattice light sheet. We're really pushing signal to noise. The movies don't look as pretty as the ones I showed you before, but each one of these big circles corresponds to a nucleus in the fly. Each one of these spots corresponds to a binding event. And what we can do is look at different, hopefully you can see the little flashing spots. Each one of those corresponds to a binding event. You can even do a maximum time projection to see where all the binding events took place, and we're gonna go back to this in a little bit. But what we can do is look at an individual binding event and measure how long the protein stays on the DNA for. You can draw a distribution, and what we see is that on average, you don't stay on the DNA for more than 1 second. And this is in stark contrast to the bursting dynamics that we measure that are on the order of 5 minutes. Okay, so already the simple model showed us that there's something that doesn't make sense, because the prediction is that bursting dynamics and binding dynamics should be the same, and here they're separated by almost 2 orders of magnitude. Okay, and what this tells you is that we need to re-examine our model, or re-examine our pathetic thinking, like Jeremy Van likes to say. And so what he likes to remind us is that models in biology are not supposed to be accurate descriptions of nature, but accurate descriptions of our pathetic thinking. They're basically ways to test our assumptions. Now, what I just showed you is that one of the assumptions in the model, assuming I didn't screw up with the math, one of the assumptions in the model is to be wrong. Okay, so I just walk you through a first round of this theory experiment dialogue, because we posited a simple model. It predicted that bursting dynamics are the same as activator dynamics. We went to the lab, we compared the two, and we saw that they didn't match each other, which means that we went through the whole cycle, and now we need to revisit our cartoon model. Okay, and of course, there are many things we could have done differently in this model. One of the main criticisms you could have drawn is that this is not the way a regulatory region in animals looks like, right? Typically, you have binding sites for many transcription factors, and those transcription factors can interact with each other. So what happens when I start thinking of this extra complexity? Well, I can posit that I have many binding sites for my activator. I can keep track of all the binding and unbinding. I can posit how that affects the rate of transcription. And I can put it all in a model where I say that the binding and unbinding is given by the timescales that I measure in the lattice light sheet microscope. Okay? Because transcription factors seem to interact on the DNA, I'm going to say that when they're bound, I'm going to introduce this protein-protein cooperativity that is classically seen in many bacterial systems. I would claim the evidence is a little bit more fuzzy in the context of mammalian, of metazoans. But the idea is that this protein-protein interaction stabilizes the binding. See how unbinding rate becomes smaller and smaller? Okay. So with reasonable values of this omega, which is in principle the only free parameter, you can use this model to simulate what the transcription rate would look like as a function of time. And what you see is that this is distributed in a bimodal fashion. Most you spend either a lot of time on or off, like you would expect in bursting. But not only that, the time scales of bursting seem to be the right ones. Even though this is 1 second, here you see that it's a few minutes per burst. Okay, so this all looks good. And if all you care about is this sort of qualitative prediction, you could have packed your bags and go home. But then you can start re-examining the model through different perspectives. And one of them is to ask, what is the distribution of activator dwell times, time that the activator spends on the DNA in my simulation? And what we see is that there's this is the predicted dwell time distribution. We see that there's a long tail of binding events that are the ones driven by this cooperativity, okay, and that are the ones that drive bursting, okay. The problem is that we never see those long-lived binding events, okay. And this is single molecule. These are single molecule measurements. I always come up with some way that we screwed up, and then I go talk to Xavier, and he convinces me that we didn't screw up, that we did all the controls we had to do, and we can talk about them. But basically, what I'm telling you is that if you look at the difference between theory and experiment, we're short, like on the order of 100 or 1,000 binding events that should be much long-lived than what we see. Okay, and those events were nowhere to be seen. Okay. So basically, what I'm telling you is that we went through a second round of this theory experiment dialogue, because I showed you that this model with a single binding site doesn't work. But even if I put multiple binding sites and I posit protein-protein cooperativity, the, the, the bursting timescales are right, are on the order of a few minutes, but the distribution of binding events not. Okay. So once again, I showed you that, even though I went through 2 rounds of this theory experiment dialogue, the simplest models that one can think of and models that are adopted throughout the literature cannot explain the bursting phenomenology. Okay. Question so far?
So there's another couple in the chat. So Larry Melidis asks, have you considered integrating genomics data either for occupancy or nascent transcription? And how do you think that would affect the model?
Yeah, so occupancy, so far, I don't know how to do occupancy with this sort of temporal resolution using genomics data. So yes, and just now I would say measurements that I believe more to have quantitative value are coming up. I'm very excited about the single molecule footprinting measurements that people are doing. No crepes are on streets, will green leaf like Ravine. So that would be very interesting to try to do it in the fly. There's some issues with needing the spatial coordinates of the cells, but still, that is not gonna give you this sort of resolution. So this is something we're thinking about, but I don't know yet how to implement it in the fly. In terms of transcriptome, transcriptomic data, it's kind of the same thing. You got very low spatial temporal resolution. These things are changing in sub-minute timescales. So that one I'm even having a harder time knowing how to go about it. Yeah.
Cool. And Claudio Franco has the question. I think he has a comment. Maybe NRA pole loading will last longer compared to transcription factor binding. And he has the question, how many RNA pole molecules can be loaded in the DNA over 1 second of transcription factor binding?
That's a great question. I think you should have an initiation event around every 3 to 5, 6 seconds. So yeah, absolutely. There's something interesting there in the sense of that's the time the polymerase also takes to leave the promoter so the next one can come on. So yeah, those timescales are very interesting, and I think they have to make us think, and that's about what the model actually is doing and predicting, and that's going to be a little bit the topic of the next part of the talk.
And an additional question from Joseph Nasser. Does the transcription factor residence time distribution intrinsically assume steady state or constant transcription factor concentrations, and are these reasonable assumptions?
We don't assume, let me think about it. No, I mean, we assume when we think about, right now I haven't showed you anything having to do with the dynamics of the concentration of the transcription factor. When we've played with that, we have assumed that it is instantaneous, there's separation of timescales such that the bursting part reads out the concentration instantaneously. We don't need to assume also that this is in equilibrium or in steady state, because we're simulating the dynamics of the process. So I hope that makes sense, Joe. All right.
Great.
Anything else?
That's all for now.
Okay, great. So those were great questions, and they are very much, they very much connect to the next stage, because basically we keep breaking the model, and that led us to re-examine our data. And what that led us to discover is that these two activators are not uniformly distributed throughout the embryo, throughout the nucleus, sorry. And when you acknowledge that that's the case, you can now think of different models that could explain the bursting phenomenology, at least in part. Okay, so, but let's take a step back. Why did the model that I showed you before work in terms of giving me the right bursting dynamics? Okay, and this actually the fact that I had a on and an off state. And the reason for that is because I had a lot of occupancy in either the full occupied state or the no occupancy state, but nothing in between. And so how do you generate these states? How do you generate, for example, this full occupancy state? Well, the classical way of doing it is that you have a transcription factor that stays on the DNA for a long period of time. And this can be realized through this protein-protein cooperativity that stabilizes the unbinding rate, that decreases the unbinding rate. But another way to generate high occupancy of a transcription factor is if many transcription factors are binding and unbinding very often, very frequently. Okay, oops. Hopefully I won't get in trouble. Okay. So, like again, if it binds and unbinds a lot. And this sort of thing, you could imagine, you could realize if, for example, you had an increase in the local concentration in the vicinity of your transcription factor binding site. So if you had more transcription factors in that vicinity, even though your transcription factors will stay on the DNA for a short period of time, very shortly after that one unbinds, the next one would bind. Okay. So what we did is we started examining this distribution of binding events throughout the embryo and inside one of the nuclei. So this is a plot that Mustafa made where you see the nuclear bicoid concentration measured in the, this is Mustafa didn't make this. This is a classic measurement of the bicoid concentration as you move along the embryo. Okay, so it's this exponential gradient. And the plot below is one that Mustafa made, which is how many detections of individual bicoids do we see per nucleus. And you see that if you're in the anterior, middle, or posterior, the mean number of detection events decreases. But then he also asked, how are these events distributed? So here I'm showing you where we found each binding event in an individual nucleus. And what you see is that these events don't seem to be uniformly distributed, but they're present in these hubs or clusters, okay? And there's something very interesting about these clusters, which is that the number of bicoid inside each one of these hubs stays the same, no matter if you're anterior, middle, or posterior. But the number of hubs changes. You have more hubs in the anterior, fewer in the middle, and even fewer in the posterior, okay? And this adds to this increasing body of evidence that, again, the nucleus is not a well-mixed bag of transcription factors. I showed you how bicoid seems to be present in clusters. We've also found clusters of this other transcription factor called dorsal. People have seen them all over the place. And it's been a little bit tricky to figure out what they're actually doing, okay? So how do these hubs change our model? Or how could they change our model? Well, once again, we have these individual binding events, we have these little clusters, and you could imagine that these clusters come about because of protein-protein interactions, okay? High avidity interactions, like the ones you find in the immune system between many transcription factors, so that, they serve one transcription factor can serve as an antenna. And now imagine that you have a transcription factor binding site there, or two, right? So one transcription factor binds, but now that can recruit other transcription factors to the adjacent site. And one way to project this onto a model is to say that instead of decreasing the unbinding rate, this sort of interaction is increasing the binding rate. Now, the interesting thing is that this is very easy to implement mathematically, and it predicts the exact same rate of transcription as you move through time, right? With the right time scale, 5 to 10 minutes, okay? But it does better. Not only does this, which is what our previous model could do, it also does better in terms of the predicted dwell time distribution of binding events on the DNA, okay? So remember, in black, this is the single molecule data, the distribution. In blue is the cooperativity model, the protein-protein cooperativity model from the previous iteration. And what I'm showing you here in green is the model that acknowledges the existence of activator hubs, okay? So this is pretty cool because it shows you, at least to the degree of the sort of measures that we could think of, we don't seem to find a way to break the model right away. Okay, so I had shown you before that the classic protein-protein cooperativity could predict bursting dynamics, but not really the binding dynamics, okay, the binding distribution. And now I introduce you to a model that if it acknowledges the presence of these hubs, and if those exist, and if a transcription factor, if a gene is inside those hubs, you could get the same bursting dynamics, but also the right dwell time distribution. I'm not saying, this is very important, I'm not saying that this is the right model, okay? But the message I want to get across is that when you have enough confidence in your theoretical models and your precision measurements, a small difference of a few tenths of seconds, a few minutes, can make all the difference between breaking a model and not, and between getting an insight about what are the mechanisms that could be at play. Okay, all right. So I showed you that what we were after is trying to endow each one of these arrows with molecular and quantitative information, trying to predict these input-output functions. But to do that, first we have to figure out what the model is of the input-output function. And what we saw is that we don't just need to measure the concentration of transcription factors throughout the embryo. We need to acknowledge that these transcription factors are present in hubs. We also, what we found is that these hubs can, if they bind to the DNA, you could imagine that they drive this transcriptional bursting and control this transcriptional bursting. And I also show you that when you put the transcription for all these bursts together, you're going to see you can get a macroscopic pattern of gene expression. And what this is doing now is this is calling again for a new iteration of our theoretical models. And we're trying to test our theory, test our understanding of what's happening here as we tune, for example, the knobs that I mentioned already earlier in the talk, such as the number placement and affinity of transcription factor binding sites. Can I use this tuning of knobs in order to shed light on the molecular mechanism of action, further light on the molecular mechanism of action of bursting, how bursts are generated and how they are controlled? Okay. We're also trying to make progress on the experimental side. And note that everything I showed you told you about binding dynamics somewhere in the genome. I didn't have a way to localize it to a specific locus. Okay, so what we're trying to do is develop technology that would allow us to simultaneously measure binding and the transcriptional dynamics at an individual locus. Okay. But also, before I finish, I just want to point out that there is a big issue with everything I showed you today. And that issue is that, I started with this huge picture of can I predict developmental programs writ large? And I showed you how we made significant progress on one specific error. And this is years and years of work. So the question is, are you going to need to do this for each one of these errors? Okay. So how can we increase the throughput of our dissections, of our quantitative dissections? And clearly, if you think about it, one of the first things in being able to predict how input-output functions are dictated by the number, placement, and affinity of transcription factor binding sites is to know the binding sites that are present at an individual gene. Okay, so if I want to start increasing the throughput of my dissections, I need to know, the first step is to know the binding site arrangement of the genes that I'm interested in. But the problem is that in flies, less than 5% of genes have any regulatory annotation. In E. coli, which you would argue is the most understood living organism, it's only 38%. Yeast, 10%. C. elegans, less than, about 0.1%. That's probably due to the terrible state of the curation of some of the databases. Okay. So what we're trying to do in the lab now is to develop techniques to be able to engage in physical biology in high throughput by leveraging DNA sequencing technology, using DNA sequencing as a biophysical tool. And the first challenge we're trying to address is to find the binding sites. Okay. And so how do you find binding sites? If you were dealing with single cells, what you would do is you would generate a library of tens of thousands of variants, like you can pay a company to do this, put this in single cells, and then correlate the expression driven by those sequences with the mutation, with the sequence, and you can find binding sites. But if I want to do this in flies, if I want to do this in multicellular organisms, I would have to inject each one of these constructs onto an individual fly and grow those flies to adulthood and keep them in my stocks. And that basically calls for making tens of thousands of transgenic organisms. And just to give you a sense, in 10 years in the lab, we've generated maybe 5,000 lines, right? So this is not doable with current technology. And these are these so-called, we can do these so-called massively parallel reporter assays in single cells, but we cannot do them in animals. So what we're doing in the lab now is inventing these multicellular massively parallel reporter assays, where what we're doing is we're recruiting DNA base editors, things that can mutate the DNA to specific regions of the DNA of the embryo, such that every embryo the mother lays has a different sequence. We can sequence that embryo and throw it away. And we've been able now, we're making great progress. We can get 40% mutation rates. I mean, it's looking pretty good. But the dream, and I'm going to stop here, is to go from complete ignorance in a regulatory sequence to finding where the binding sites are, to figure out who's binding there, to mapping the gene regulatory networks, and controlling and predicting those gene regulatory networks. So that's one of the big challenges that we're excited about coming up. So to wrap up, let me just remind you that this is the great work of Megan and Nick in my lab with the help of Augusto, Emma, and Simon, and also the result of a fantastic collaboration with Chris Wiggins, Mike Eisen, and Xavier Dazac. Let me also acknowledge my lab that has, we've gone, especially after COVID, we went through a full round of a turnover of students and postdocs, my great collaborators, my book co-authors, an amazing group of people to do science with, these funding sources that give us the freedom to do the science that we're interested in, and all of you for your attention and your questions, and I'll be happy to take more questions. Thank you very much. Thank you for having me here.
All right, thanks very much, Hernan. That was a very inspiring talk and kind of a beautiful interplay between theory and experiment. So I see we've already got some questions in the chat. So if anyone else has questions, put up your hand or put them in the chat, and I'll start with the questions in the chat. So related to the transcription factor hubs section, Fernando Caceres says, if there are 2 alleles per locus, but several greater than much greater than 2 transcriptional hubs, it is the alleles, the chromatin, that should enter and exit the hubs. Now the kinetics of chromatin loop movement needs to be included in the model.
Yeah, I agree. I still don't know, but honestly, I didn't even show you that there's there's DNA, like the locus that I care about is in that hub. So I think that's the sort of thing that we need to think about. And it's true that the chromatin moves at a slower dynamics than the binding and unbinding, and these hubs are transient too. So yeah, I agree. We need to do that, and I think again, this needs to be incorporated in the model. You're absolutely right, but we also need better measurements. And people like Mustafa now in his lab at Penn, he's making good progress in that direction, I think, as well.
And Ruta asks, does the existence of these hubs of transcription factors affect their diffusion in any way? And she's wondering whether some of the bound states observed from FCS could correspond to these hubs, and perhaps that's why occupancy times did not match between theory and experiment.
Yeah, that would be interesting to be able to do FCS on the hubs. Ooh, that would be hard right now, I think, with the current technology. Yeah, I don't know how to do that right now. Yeah, no, it's a great question. It's a great question. It's just that remember these hubs seem to be defined over time, like it's the accumulation of binding events. So it's not a structure like for now. I don't think it's defined as a structure. The dorsal clusters that I showed you on the right, those seem to be more stable, for example. And I can tell you that those dorsal clusters like to be closer to the DNA that they regulate. So this is dorsal regulating, this is the distance between dorsal and SNIL, that is a target gene. And you can see that it likes to be closer to it than hunchback, that is a non-target gene. This is the distribution of distances between clusters and the gene. But yeah, the diffusion, that's a great question. That's a very good question. I don't have a good handle on that right now.
So Edouard Tordeau asks, how would you hypothesize that in time and space, protein complexes can meet inside nuclei crowded with chromatin mesh and precede such synchronized events of transcription? And he noted that especially because of the diffusion might be strongly reduced by protein size.
Yeah, that's a good question. And clearly some of this has happened, right? And, even though we have this picture of the nucleus and the cytoplasm being so crowded, things can move around. But I think the question about synchronization is a very good one. So one of the things we're trying to do now is, everything I showed you was with a heterozygote flight, which has only one copy of the reported gene. But you can do it with 2 copies of the gene, right? So now you can compare how well synchronized the two alleles are. And you might be able to constrain some models related to the questions that you were asking in that fashion. That's one of the hopes we have.
And Olivia asks, what about a hit and run model where each different transcription factor leaves a mark?
Yeah, I mean, this sort of thing is stuff that we want to be able to do, but you need to have some way of measuring the concentration or the binding in the same locus that you're looking at, and that's been a little bit of a challenge. Let me see if I can find. So, one of the ways we're going about it is, inspired by great measurements by Ido Golding and also, Ibrahim Cissé did some stuff, and Jun Ma, people, Ido has, for example, asked the question of, is there more protein in the vicinity of the transcription factor binding site of the gene than anywhere else in the nucleus? They did this in fixed tissue, and they saw that indeed there is more protein here, and that's related to binding. So one of the things we're trying to do is, well, can you do this in real time? Okay, so for example, this is dorsal in green in the vicinity of SNIL, which is a gene in magenta. And if you look at dorsal concentration in that vicinity, you see nothing. Like you see no signal. You can look at different snapshots, you see nothing. But when you start averaging over multiple snapshots, you start seeing that there's an enrichment of dorsal in the vicinity of the gene. And that seems to, according to Ito, seems to be related to binding dynamics. So then the sorts of things that you could ask in the sort of kiss and run thing is, if I measure dorsal concentration at the locus as a function of time, and I compare, in a control where dorsal is not supposed to bind, and I measure it in the gene itself, are these transient fluctuations, these transient increases related to binding events, and are they related to bursting? So these are the sort of questions that we want to ask. And up until now, we were very, very limited in signal to noise, so we couldn't ask this. And now we're making great progress both on the transgenic technology and the microscopy technology. And I think we might be able to answer those questions of whether the transcription factor needs to be there for the whole duration.
Cool, that's very interesting. So Malika Panja asks, how do these hubs change with the stage of development?
We only measure them in cycle 14 and I think in 13. So I don't know the answer. But Bitcoin doesn't stay around much longer. And it can't drive much productive transcription before 13 because the nucleocycles are too short. So I think this might be it for the Bitcoin phenomenology. That's my hunch.
Mm-hmm. And Claudio Franco has another question. Do you think that transcription factor hubs is a common feature of transcriptional regulation, or is there a specific pattern or is it specific to a pattern of a transcriptional burst?
No, I mean, people have been doing screens and they keep seeing, I don't know if it's a hub in the way that we define it here, but they keep finding transcription factors that are not uniformly distributed throughout the nucleus. So this seems to be quite a widespread feature. I don't think it's necessarily true. And there's different time scales that might be associated with each cluster or hub. But to me, I think I wouldn't be surprised if many of the transcription factors we'll be looking at behave in this fashion.
So Ulrich Kaiser has a question. Is there any chance of transcription factor self-quenching if more than one transcription factor binds, or is it associated to the hubs?
Self-quenching, I guess, in the sense of excluding each other or something like that, or
I think so.
Yeah, I guess, yeah. I mean, if it's excluding each other from the DNA, that would have to do with the DNA geometry. The quenching, in some sense, well, another way to think about the quenching is like, imagine that another explanation for the hubs is that instead of delivering transcription factors to the DNA, they actually try to take it away from the DNA. It could be that the target gene is not in the hubs. And I guess that would be a definition of quenching. I don't know if you want to mute yourself and clarify the question, but that's the sort of thing the way I would answer the question as I understand it.
So yeah, just to clarify, so if you had one model where you had more than one transcription factor in the vicinity of the DNA, yes, they could also interact with each other, no? So the dye is on it. Does that make sense?
What would I mean, in the simplest model that accounts for the hubs, once the transcription factors are bound, they don't interact with each other. They just interact with the machinery. But you could imagine that you add also protein-protein cooperativity to that model.
This is my question. Yes, is there, because I understand that this is the simplest model you have, but I mean, in the end, you are in a very crowded system. Yes, I mean, you have a lot of interacting.
But what do you mean by quenching? Just tell me what quenching means in this context.
But if you have dyes very close together, yes, because you have labels, they can self-quench. Yes, you can have other, and then they actually disappear while they are still there, and you can't see them anymore, but they disappear from the measurement.
Sorry, now you're talking about a detection artifact, not a functional thing.
No, but it can be either. Yes, I mean, there's
got it. Yeah, I don't think there's enough quenching, as far as I can tell, with GFP for it to make such a difference.
Okay, thank you.
People do this to find they use GFP. They look at steps of GFP in diffraction limited spots when you have more than one GFP.
Okay, thank you.
So I would be surprised if that's the case. You can design systems that have quenching, but I don't think GFP would have that sort of behavior.
Okay.
So it's 5 o'clock, so let's just ask the final question in the chat. So Samir Mirabat says, it looks like that your hub connected model relates more to high transcriptional efficiency. And what about weakly expressed genes?
That's a great question. Well, I don't know, because weakly expressed genes, sometimes the way we think about it is that the promoter, you know, it depends on what a weakly expression is. Is it that it bursts just as often, but with a lower amplitude? Or it's like very high amplitude, but very seldom transcription? So it's not clear that the definition of weakly, like there are multiple ways of realizing a weakly expressed gene, you know. And so in this context, I think these hubs are controlling the onset of the bursting, so the burst frequency to some degree. So, you know, then you would, I guess, in this sort of thing, you would demand the same kind of burst frequency for weakly and strongly expressed genes. So maybe in that case, you would have, in the same context, assuming that a weakly expressed gene works in that way, I guess the way to realize it would be with a weak promoter. And people like Munialaga have looked at what the promoter sequence does to bursting, and I think it is indeed changing mostly the amplitude.
Great. All right. Thank you, Hernan, for the great talk again. And thanks everyone for joining us today.
Yeah.
Right.
If anybody has more questions, just shoot me an email and we can talk.
Okay, great.
Nice. Bye, guys. Thanks for having me.